Okay, so this is just a quick review of Chapter 4 from the Pierce textbook. And we want to talk about sex determination, sex linkage, and sex determination. We want to look in humans and flies. That leads to the idea of dosage compensation. Um, let's see. And I think that's about it. This also uh, leads into a little bit of what's called the chromosome theory of inheritance, meaning that genes are on chromosomes. We know that. But the fact that um, X-linked genes, the discovery of X-linked genes led us to this, and simply stated that just means genes are on chromosomes. We don't have to go into a whole lot of the history of that, but that's kind of the main points of this chapter. So so let's start out with sex determination, and we'll start out in humans. So this is sex determination. I think you all know that if you're an XX individual in humans, you're female, and if you're an XY, you're male. So what determines sex in humans then? Is it the number of X chromosomes? Maybe, but there are a couple of sort of rare situations where we can have just at one X and an O, and we're still a female. That happens to be called Turner syndrome. And while there's various characteristics of this syndrome, the underlying importance for this is that it's a female. And if you have two X's and a Y, you're a male, and that's called Klinefelter syndrome. All right, so that's sex determination in humans. And what this tells us is that the Y determines maleness in humans, or male sex in humans. And so we're interested in what the actual evidence for that is. And the evidence is that if we take the Y chromosome away, we get a female. All right. So just to contrast that to fruit flies, just to show that there's different methods of sex determination. XX is a female, XY is a male, but XO is also a male, and XXY is a female. And so that means it's the number of X chromosomes determines sex. It's a little bit more complicated than that, but well, it's kind of a lot more complicated than that. But um, if you have two X chromosomes, you're female. One X, you're a male. And that's basically how it works in flies. So it's different than humans. And I'm not covering every detail of this. This is just to cover the most important points. So this leads to the idea of dosage compensation. And we'll worry about this mainly in humans, which is more broadly in mammals. Dosage compensation means if we have an XX individual or an XY individual, so that's a female and a male. So just thinking about the normal situation, um, the amount of X-linked gene product So however you want to indicate this, so X-linked gene product, that means how much RNA and protein do we express from the X chromosome. Because females have two X chromosomes and males have one X chromosome, you might think that there's two times as much X-linked gene product, but there's really the same amount. So whatever it is, we'll just say it's at a level of 100%. You're supposed to, whatever it is, the amount is equal. So how is it that in females that have twice as many X chromosomes, we get the same amount of gene product? In males, we've only got one X chromosome, we get the same amount of gene product. So there's different ways to do that. We would expect females to have more X-linked gene product, but they don't. So the way it works in mammals is that in females, during development, you have a cell, and there's two X chromosomes. That cell divides, and in each cell, at some point, one of those X chromosomes gets inactivated. And so, in this case, this this X chromosome is being inactivated. And in this case, the other chromosome gets inactivated. So that gets inactivated. And the fact that I drew the arrows going both ways, that's just because I happened to put those little blobs right there. It's random which of the X chromosomes in each cell gets inactivated. So this process is called X inactivation. This thing here is called a bar body. And this is a mechanism of dosage compensation in humans. And so it's compensating for the fact that there are two X chromosomes in females. So it's reducing the amount of X-linked gene activity in half by getting rid of one of those X chromosomes. But it's random. So then when the female grows up, and we'll do this for a cat. So this is either a cat or maybe a turtle. Turtle's not a mammal. I'm just saying it looks like a turtle. So let's pretend this is a cat. Give some cat-looking ears. There's some legs. In cats or in any kind of mammal, if you look at, you can have different patches of tissue that have a different X chromosome inactivated. So these guys could have this chromosome inactivated, and this part of the cat could have the other X chromosome inactivated. And so that means all the genes that are on that inactive chromosome are turned off. And so if there's a gene on this X chromosome, and there's a gene for orangeness. I don't know if I have orange as a color. Let's just pretend red is orange. So this guy might be orange, and this area might be black. And so when you see a calico cat that has this kind of pattern, the reason is, or at least one of the reasons, is that different patches of the tissue, different patches of the skin, in this case, or the hair, has a different X chromosome inactivated. And if there's a gene on there that you can tell what's going on, you'll get different phenotype just in those different patches. That same thing happens in humans. All right, so now I got to draw a human. My drawing's not so good. All right, so we're going to draw a human. All right, there's a human. All right, that looks kind of like a human. And it's female human. And female humans also have different patches of skin that So in humans, the same thing happens. But so you'll have a patch of skin that has one gene turned on and another patch with the gene turned off. So you might have um, this patch of skin has one of the X chromosomes inactivated. This patch of skin has a different X chromosome inactivated. And there's a gene in this case. I can't remember the name of the gene exactly, but there's a gene that results in sweat glands that are present in one patch of skin and no sweat glands in the other patch of skin. And so it turns out that human females, cat females are all what we call mosaics because they have different X-linked genes being expressed in different parts of the body. And so that's one of the consequences of X inactivation, which again is X inactivation is a mechanism of dosage compensation. We'll come back to this because this process of going from an X chromosome down to a bar body, that's a very important change in chromatin structure. So the chromatin structure gets condensed. Chromatin is the material that makes up chromosomes, and so we'll talk about that more later. All right, so now X linkage, or sex linkage. You've all done this already, and so we can do a quick review. And the first thing you need to be able to do is to recognize the inheritance pattern that leads to sex linkage. And let's pick, well, we'll go ahead and use the white gene. I guess we're using the green pen for the white gene. And if you take this was where it was originally discovered in fruit flies. And this was Calvin Bridges and Thomas Hunt Morgan. And it's the 100th anniversary. This year is the 100th anniversary of this discovery. And the basic observation was that if you did the cross in this particular way, you got a difference in the sexes. So and this is white eyes. So you got white-eyed males and red-eyed females. And so that means that this trait is somehow linked with sex. All of the other traits that we've looked at so far have been autosomal. So autosomal means not sex-linked. All right. Now, what Bridges did is he could actually look at these chromosomes. And he knew that you can actually see the chromosomes in the females and in the males. And males have this little Y chromosome that actually kind of looks like this. So sometimes I'll end up using that for the Y chromosome, but we can use a Y chromosome. And so what Bridges did is he said, well, maybe if the white gene is on the X chromosome and the wild type version of the white gene is on the other X chromosome, then we could explain this inheritance pattern. And here I'm just going to use plus for the wild type version of white, and the plus is dominant. And so this is sex-linked recessive inheritance. I'm not sure what's up with this pen. And what it does is we know that we get one X chromosome from mom. And if you're a male, you have to get the Y chromosome. And then you can get an X chromosome from mom to be a female, but you have to get the X chromosome from dad over here, and that's got the wild type allele, and so it's dominant. So the first thing to know about sex linkage is that if you set the cross up in this way, mutant female cross to normal male, so male, all sons are mutant, and daughters are normal. And so that's why in the virtual fly lab, I always have you set up with the mutant female and the wild type male, because um, the sex linkage shows up right away. If these were all red, then all we could say is white is recessive and it's autosomal. Now, if you do the cross the other way, so we have to do the reciprocal cross. So what we just did was take a white female and cross it to a red male, and we got white males and red females. Sometimes I've heard people call this a what do they call it? A crisscross pattern of inheritance, where we started out with a white female and now we switched it so that the males um, are now the have the mutant phenotype. But anyways, so what we just said was the females had the white gene, the white allele. The males had the wild type allele. So down here, all the males get an X chromosome from mom and a Y chromosome from dad, and the females get an X chromosome from mom and an X chromosome from dad, and they look like this. And that's the F1. So if we set up the cross in this direction, we see the result of sex linkage in the first generation. If we do the reciprocal cross, now we're going to take a red female, cross that to a white male, so it's the opposite direction or the opposite way to set up the cross, and we call that reciprocal. I don't have room to write it right now, but and so here now we're going to get all wild type, which is the same as red. And why is that? It's because we have now this female, assuming she, in this case, she's true breeding, means she's homozygous for that trait. The male is white. We know he's got to have that genotype because white is recessive, and he's a male, he only gets one X chromosome, so it has to be that. So now the females are going to be red, but they're going to look like this, and the males are going to be red and they look like this. So here we can't tell. So if we just did this cross on the right, we wouldn't be able to tell if it's sex linked or not. If this was autosomal, we'd get the same result. We cross a red to a white. If red's dominant, we'd get all red in the F1. We cross those together in the F2, we would get 3 to 1 with no difference in sex. But here on the left, if we just stop at the F1 with both these crosses, because we see a difference in the reciprocal crosses, because these are different, that tells us that they're sex linked. If the reciprocal cross matters, that means they're sex linked. But then we can look at what happens with this guy. We can see in the next generation, we can get X plus X plus. So we can get this chromosome and this chromosome. We can get X plus Y. So that's getting this chromosome and this chromosome. We can get XWX plus. It's getting this chromosome and this chromosome. We can get XWY, where we're getting this chromosome and this chromosome. And so what do we get? We get a red female, a red male, a red female, and a white male. Alright, so now this is the F2. When we do the cross in this direction, we see the difference in the F2. If we cross these guys together, we get XW, XW, XWX plus XW. So now we're getting this one and this one, and Y, and X plus and Y. So we're getting that one and that one. These are white. These are red. And so in this case we get half red, half white, with no difference in sex, but it's different than a monohybrid cross. So we're not getting 3 to 1. This one we've got, it's kind of a 3 to 1 ratio if you ignore the sex, because we get 3 reds and 1 white, but if you pay attention to the sex, we get, if we combine them, we get 1/2. Let's see, so we get 1/4 of each of these. So I'm going to put this on the next page. So in that case, we got so just to do it again, if we took a now we're taking a red female across to a white male, we get all wild type. Make those together, what we get is 1/2 red females, 1/4 red males, and 1/4 white males. And you can fill in the chromosomes to show how that works. And it would look like. So pedigrees, we have a circle and a square. And the circle's female, and the square is the male. If it's filled in, it's affected, not infected. So you always want to say affected. So if it's an X-linked trait, it's affected with that trait. Infected means that there's a bacterium or something, or a virus that's infecting you. Affected means you're affected with a genetic trait. And then when we do a pedigree, we get some kids out of this little matrimony here. Get some boys and some girls. We can number our generations like this, and we can number our people like this. That just kind of helps you keep track of who you're talking about. So I could be talking about 2.4, and in this case that's a male. But if it's a sex-linked trait, all the sons will get it if the mother has it. So that's one way to recognize linkage or sex linkage. But you don't always see it that way, because recessive traits tend to skip a generation. So you could see something like this. Maybe you'll see something like that. Now this isn't a whole lot of information to make any hardcore conclusions, but we can conclude a couple of things. If we see a pedigree like that, we can say immediately that whatever this gene is it's recessive. Because anytime that you see two unaffected individuals having a child that's affected, it has to be recessive, right? Because if it was dominant, then one of these parents would have to also be affected. Now, if you read through, all of genetics, that has, there are other possibilities for like different expressivity and penetrance and things like that. But right now, we're assuming that these things are completely penetrant, normal traits. That if you get the gene, you get the you get the disease or the trait or whatever it is. So this certainly could be autosomal recessive or X-linked recessive, but when you start to see only males get it, or males getting it much more than females, then it looks like it's sex-linked, and it's sex-linked recessive. So that would tell us that this original, let's say it's color blindness, that's a common one. That female would have been heterozygous for the trait. This male would have a Y chromosome, and he'd have to be normal because he's unaffected. Then all of these boys must have got the big C from or these girls. Well, these girls could be, we don't know if they're carriers or not yet. So the girls could get either X chromosome, so we don't know what's going on. But we know the boys get the Y chromosome, and if they're unaffected, that's a big C. This guy is coming in, marrying in from outside of the family, which is usually a good thing, and he has a big C as well. Now, half of her sons have it, and so that means she must have got the little c from her mom and the big C from dad. So 50% of her kids are going to get the little c. And if they get a little c and a Y, then they get they're colorblind. And if they get the big C and Y, they're normal. These may or may not have the big C or the little c. We don't know yet. So that's when you're doing pedigrees. The first thing you do is you look to see if it's dominant or recessive. Anytime you see where everything, you might see something like this where you never get where the parent, if somebody's affected, their parents were always affected, and you never see something like this. So I guess if you see this, you can rule out dominant inheritance. But if you never see that and you get the trait in every generation, then it's probably dominant. And we can have X-linked dominant traits. So if you have an X-linked dominant trait, so we'll just give it a big D, so because it's going to be dominant, whatever it is. That means if a female has it, she's probably heterozygous because most of the time these traits are rare, and if it's a dominant trait, you're not homozygous for it. And then so she'd have to give it to, if it's X-linked dominant, you can't really tell a whole lot. Throw a male down here, but she could give it to. I mean, if it's X-linked dominant, and so she's heterozygous, so she has this genotype with a little d, and she's heterozygous for it. Then she can basically give it to her kids in about a 50-50 ratio. But if this guy gets it, he would have to pass it on to all of his daughters because he's got X big D Y, and all of his daughters have to get his big D and then an X from mom, X big D. So that's X-linked dominant. And the diagnostic thing is males pass the trait to all daughters and no sons, right? So that person doesn't have it. So if she doesn't have it and he does, all of his sons have to get the little d from mom and the Y from dad. So pedigrees take a little bit of practice, and they're not, sometimes you can't really say for sure, but I'll do a separate little thing on pedigrees.